Hi everyone. Today I am going to talk about the complement system. What is the complement system? It is a defensive system. It consists of over 30 proteins and it's produced by the liver and is found circulating in blood, serum and within tissues throughout the body. The complement system is so named because it helps the cells of the immune system in destroying the microbes. The complement system is not adaptable, that is, it does not change over the course of person's lifetime and it is thus belong to the innate immune system. However, it can be recruited and brought into action by the adaptive immune system. The proteins of the complement destroy microbes by cytolysis, inflammation, or phagocytosis and thus prevent excessive damage to the host tissues. There are three biological pathways that activate the complement system. The first one is the classical complement pathway, second is the lectin pathway, and the third is the alternative complement. First, we are going to discuss about the classical pathway. In the classical pathway, the antigen antibody immune complex, thus formed, produces conformational changes in the Fc portion of the antibody molecule. This exposes the binding site for the C1 component of the complement. The C1q then binds to the Fc region of the antigen complexed antibody that induces conformational change in the C1R molecule. That C1R molecule then converts into serine protease that activates C1s molecule. The structure of C1 complex. C1 complex exists as a macromolecular complex and it consists of one molecule of C1q and two molecules each of C1R and C1s that acts as serine proteases. These are held together by calcium ions. The C1s molecule can act both on C4 and C2 molecule. C4 molecule is activated when C1s hydrolyzes it and it cleaves into C4a and C4b. The C4a is the smaller fragment and it acts as an anaphylatoxin. The C4b attaches covalently to the target membrane surface in the vicinity of the C1 where it binds to the C2. When it binds to the C2 becomes susceptible to the cleavage and it cleaves into C2b, a smaller fragment that diffuses away, and the C2a, the larger fragment. The C2a binds to C4b, the complex C4b2a. This C4b2a complex is also known as C3 convertase. It remains membrane bound and hydrolyzes C3 into C3a, the smaller fragment which acts as an anaphylatoxin, C3b. The C3b then acts in three ways. The C3b can bind covalently to microbial surface where it acts as a molecular tag, and this is recognized by phagocytic cells with C3b receptors by the process of opsonization. The C3b may attach to soluble Fc portion of antibodies in antigen antibody complex, and these C3b tagged immune complexes are then recognized by C3b receptors on the phagocytes or RBCs. The C3b can bind to C4-2a enzyme forming the C4-2a-3b C3 convertase, and this acts as an C5 convertase complex. The second pathway is the lectin pathway. The lectin pathway, like the classical pathway, proceeds through the activation of C3 convertase composed of C4b and C2a. However, instead of relying on antibodies, this pathway uses lectins as its specific receptor molecules. The lectins are proteins that recognize specific carbohydrate components that are primarily found on the microbial surfaces. One example of this lectin protein is mannose binding lectin protein, MBL. MBL is associated in the serum with MBL associated serine proteases or MSP proteins. There are three MSP proteins that have been identified till now: MASP1, MASP2 and MASP3. MASP2 is structurally related to the serine protease C1s and thus can cleave both C2 and C4, giving rise to the C3 convertase C4b2A. Thus, the lectin pathway utilizes all the same components as the classical pathway except of the C1 complex. MASP proteins takes the place of C1R and C1S in cleaving and activating the C3 convertase. Once the C3 convertase is formed, the reactions of the lectin pathway are similar to that of the classical pathway. The lectin pathway. It uses lectins as receptor molecules, for example, mannose binding lectin. The MBL forms complex with the MASP2 protein and it functions similar to C1 complex. It cleaves C2 and C4 and forms C3 convertase C4b2A and then it follows the similar pathway as classical pathway. The third pathway is the alternative pathway. In this, the C3 undergoes spontaneous hydrolysis to form C3H2O, which binds to serum factor B, C3H2OB, that is cleaved by serum factor D into C3b, which is released, or C3HBB. The released C3b then binds to the microbial surface and it further binds to factor B, thus becoming susceptible to cleavage by factor D and forming C3 convertase C3bBB. This C3bBB is unstable and is stabilized by the protein properdin. This properdin stabilized C3 convertase then binds to C3b fragment and forms C5 convertase C3bB. The C3H2OBB also binds to C3b fragment and this forms the C5 convertase C3bB. The three complement pathways then converge at the formation of the C5 convertase. The C5 convertase in the complement pathway, classical pathway or the lectin pathway is C4b2A3B, while in the alternate pathway it is C3bBB C3B. This C5 convertase cleaves C5 into C5A, the labile portion, and the C5, the larger fragment. The C5b then binds to C6, that further binds to C7, and then it forms C5b6-7 complex. This then binds to the C8 beta part of the dimer C8. C8 beta part of the dimer C8 then either binds to C5b6-7 forming C5b6-7-8 complex that binds and polymerizes with C9 to form MAC complex, or it undergoes conformational change forming C8 alpha gamma that binds to phospholipid membrane and creates small pores of around 10 degree angstrom and that can cause lysis to our MAC complex. It consists of C5b6-7-8 complex surrounded by poly C9 complex, resulting in a tubular structure that can cause functional pores of diameter of around 70 degree angstrom to 100 degree angstrom that results in the loss of the plasma membrane integrity, leading to the irreversible cell death.